my name is Ismail Zulaika, and today I'm going to talk about comparative genomics of Mycoplasma pneumonia isolated from children with pneumonia.
So, for introduction part, pneumonia is an important cause of respiratory tract infections in children and adults. So, this is the picture of pneumonia, and also this is one of the manifestation that is included, which is skin rash. Next, P1 adhesion mediates cytoadherence to the surface of respiratory epithelial cell. So in order to determine the genotype, P1 typing is used, but it did not convey information regarding clinical severity. So new genetic technique is used, which is MLST and MLVA. So despite evolution, research to understand the entire genome structure in regard of molecular biology is still left behind. So in recent advances, it is possible to analyze pneumonia through high-put sequence technology, such as Illumina dye sequencing. So the objective of this study is to analyze genome of 30 pneumonia strains isolated from children with pneumonia in South Korea during the two epidemics, and also compare it with global connection strains, which include 7 countries. Its methodology. So first is pneumonia strain. So it will be detected from children with pneumonia in South Korea. Next is cultivation. So it will be performed using pneumonia-like organism broth, and strain M129 will be used as a reference. Next is DNA preparation. So it will be extracted directly from the cultivated pneumonia using extraction kit, and MLST analysis and P1 typing will be done. The next one is selection of strain for whole genome sequencing. So 30 strains were chosen. And then next generation sequencing, there is a 4 basic steps for NGS, which is library preparation, cluster amplification, sequencing, and alignment. So instead of directly aligning the reads to a reference sequence, de novo assembly was performed. Genome assembly and annotation. So NGS reads were assembled de novo using spades, and then the initial NGS reads were aligned to de novo assembled genome for correction of errors. So annotation will also be done by using rapid annotation subsystem technology, which is RUST. And then comparative genome will be done to detect large chromosomal rearrangement, deletion, and duplication. MALF will be used, and to construct phylogenetic tree, Philo-IO and Mega-X will be used. So single nucleotide polymorphism, and then next is protein functional analysis. Patrick was used, and heat map was generated based on annotation. Next is the results obtained. So first, stain characteristics. So the stain were isolated from nasopharyngeal samples obtained from children with pneumonia. And then genome assembly. So after the assembly is done, genome will undergo automated gene annotation. The next one will be overall comparison. So based on the picture, as we can see, solid coloration indicates more than 99% identity, and transparent gray indicates approximately 95% identity. So the genome were 99 to more than 99% identical, and similarities will drop to approximately 95% in the gray area. And then for genomic structure comparison, mouth detected 4 subtype specific insertion, and the insertion can be seen in the picture. So next is SNP and indel analysis. So they show fewer variant numbers in P1 type 1 strain and then compared to P1 type 2 strain. So for protein and functional analysis, based on the picture, heat map viewer is providing a comprehensive view of the distribution of the protein families. So there are distinction between the genome, and it was apparent. So phylogenetic association, this is the phylogenetic that they built. So they was divided into 2 clade in accordance with P1 typing. And as we can see, clade 1 form the largest clade overall, and also shows the most heterogeneity in terms of both origin and time of strain collected. Pneumonia is an organism that is difficult to culture, so the aid of molecular biology is critical. So despite the effort of P1 typing, it is not sufficient. So they use recent advances in technologies such as MLVA and MLST. They also use high throughput technology, but not extensively applied. The study concluded that pneumonia is extraordinarily stable over time and geographical distance across the globe. However, there are limitations, such as number of strains included are small, and then isolates were chosen from 2 consecutive outbreaks only. But nevertheless, this study expanded our understanding of the genome structure of pneumonia through whole genome analysis.
The conclusion would be the comparative whole genome approach was able to define high genetic identity, unique structure, diversity, and also phylogenetic association among the 78 strains isolated worldwide.